Hi everyone. Well, it seems like I have my camera back, so that's a good thing. So welcome to you week 3 of your journey into the world of the unseen. Now, it might seem strange for many to think of microorganisms, and especially unicellular bacteria, as living organisms, which are, at least on one level, no different from humans. I mean, the very basic biological purpose of the human, or any animal for that matter, is to propagate the species. Likewise, a very basic biological purpose of bacterium is to propagate the species. Now, bacteria respire, they ingest nutrients, they metabolize those nutrients, they excrete waste products, and they reproduce just like humans. The point is, in order to do all of that living stuff, bacteria need genes, well, just like humans. Now, bacteria carry their genes on a single circular chromosome. Humans have 23, or two pairs of 23, 46 total, and our chromosomes are linear. And so this week we take a look at microbial metabolism and bacterial genetics. Then we'll examine first how bacteria can manufacture the energy necessary to do all of that living stuff. And second, how they can transfer characteristics, for example, like antibiotic resistance, to their offspring. We call the offspring progeny. I have provided links to 3 videos to help you understand the concepts here in week 3. The first is a video describing the process of aerobic and anaerobic respiration. Remember, aerobic means with oxygen or in the presence of oxygen. Anytime we put an on the front of a word, anaerobic, that means without oxygen. There are bacteria that can actually function and survive well in the absence of oxygen. So the second explains the central dogma. We call it the central dogma of molecular biology, and it describes the flow of genetic information from DNA to RNA to protein. And every living organism plays by that playbook. We call it the central dogma. Whether you are a human, a pine tree, an elephant, or a bacterium, we all play the central dogma plan. And the third explains methods of gene transfer between bacteria. Now, you're familiar with the idea of binary fission, right? That's where one parent cell physically divides into two daughter cells, and those daughter cells are genetic clones of the parent. And so if you have one parent dividing into clones, and then they divide into clones, and everybody is the same genetically, then your question may come to your mind: how does variation occur within bacteria? Because after all, it's variation that is the fuel for natural selection. So where do you get variation? We know where you get variation, for instance, in humans, right? Mom and dad, you get chromosomes from mom, chromosomes from dad, and they mix, and that's where you get variation from. But what about bacteria? Well, bacteria are able to transfer genes between themselves, um, between them and other bacteria, and they're able also to pick up just loose strands of DNA floating around in the milieu, in the environment. They can pick those up, and the third video will explain those processes for you. Okay, so we're talking about in chapter 6 microbial metabolism. And so all of life is dependent upon a constant supply of energy. Now, the penultimate source of energy, of course, is the sun for all life. You are familiar, I'm certain, with the process of photosynthesis. Photosynthesis process where plants and algae, for instance, and some photosynthetic bacteria, they absorb light energy from the sun and convert that into chemical energy, which then is used by the rest of us to make our energy and to survive. And so you'll learn about that chemical energy. Now, chemical energy, we call it ATP for short. It's adenosine triphosphate, and you probably remember that from 10th grade biology. And so chapter 6 describes how bacteria are able to harness ATP to do their living stuff. Okay, chapter 7 is a blueprint of life. This is the central dogma that I was speaking of before. Now, the genome is the sum of all genes that make you who you are. And everything that you are, do, think, and feel is controlled by your genes. Our genomes are literally wrapped up in those things we call chromosomes. And our chromosomes are made of DNA. And again, the central dogma describes how those genes and DNA are turned into the stuff that is you. And so you'll learn about DNA and RNA and genes and proteins and how the central dogma is true for both eukaryotes like humans and the microorganisms like bacteria. And then chapter 8 is all about bacterial genetics. As much as we humans try to maintain stability and familiarity within our environments, the fact is our environments are always changing. They're forever in flux. So we don't usually even notice the changes unless, like, I'm on the East Coast and we almost literally go from cold winter to hot summer. We have a little bit of a spring in there, but not very much. And so here we notice the cold to the heat, and we notice when it goes from uh, 85 degrees to 105 degrees here on the East Coast. But usually, humans, we don't notice those kinds of changes in our environments. Nevertheless, our environments are always changing, they're always in flux. Of course, if there's some natural disaster, by the way, I just heard about an earthquake in Colombia, so be praying for our brothers and sisters down there. But something like that happens, we notice the change. But normally we don't even know. If it changes 2 or 3 or 5 degrees, we don't notice the change. But our body does, and our body will make adjustments to those kinds of things. Well, the environments of bacteria are also in flux, always changing, and they change in major ways for bacteria. We don't always notice the changes, but bacteria notice the changes, and they're constantly adjusting to those changes in order to survive and reproduce and propagate the species. Okay, so you'll learn about mutations. That's the fuel for natural selection, as I mentioned, which allows bacteria to evolve, and they evolve very rapidly. Remember that the generation time for E. coli, for instance, is only about 20 minutes. So every 20 minutes, the population doubles. So mutations or evolution within populations of bacteria happens very quickly. Okay, housekeeping items for the week. Make your discussion post early in the week. Some of you are still waiting until Friday evening. They're due at 11:59. Some of you are still waiting until about 11:50 on Friday night to do your initial post. And my note to you is always post early in the week so you can fully engage in the discussion process. If you post late Friday evening, that really doesn't give your peers enough time to read and process your post and then properly respond. Also, some of you like to pose follow-up questions in your post, which is a great thing. It helps to keep the conversation going. However, do not wait until Saturday evening or Sunday evening to add follow-up questions. By that time, everybody else is through with discussion. It's a moot point for you to add a follow-up question when you're doing your post Friday night, or when you're doing your follow-up, your peer responses on Sunday evening. Right? It's a moot point to do a follow-up question. You don't get any extra credit. You do not get any additional credit for follow-up questions. If you're posting earlier in the week, follow-up questions are great. Saturday, Sunday evening, not so much. Your topic 3 posts due no later than Friday. That's August 14 coming up, 11:59 p.m. GCU time. There's a microbial metabolism and genetics homework assignment and also quiz. Both are due no later than Sunday, August 16, 11:59 p.m. GCU time. Remember to check class resources. I add helpful things there all the time. Next week is the midterm, and you will have a midterm exam, and I will post a topics review for your midterm exam in class resources. So if you're not checking class resources, you're missing a lot of stuff. And some of you are not, because I can see in your discussion post you have not reviewed my guidelines for discussion posts, which is a PDF located in class resources, and this is not the first time that I've mentioned that. So make sure you check those resources out on a regular basis. Okay. Pay particular attention to due dates on all of your assignments. Proper credit can only be given for complete and timely submissions. Any questions, issues, problems, contact me as soon as possible. Message 804-229-7993. You can contact me on Halo Message or Inbox, whichever term you like there. Those are the two quickest ways to get a hold of me. And then certainly you can use snail email. I'll get back to you within 24 hours. If I'm in my office and you message me or message me through Halo, I will usually respond to you right away. Okay, free animated education, aerobic and anaerobic respiration, the video, biocast, central dogma, the video, and the video from USMLE, transformation, conjugation, transposition, and transduction. Those are the ways that bacteria can transfer genes between them and generate mutations for natural selection. Okay. Let me know what's going on. Oh, yeah, let me say this. It's not in my written announcement, but it probably will be. I'll put it in there next week. But you have coming up at the end of the term, you have a PowerPoint or a slide presentation to do on a disease or a disease pathogen combination. Those topics are pre-assigned to you, so you need to message me via Halo to inquire of your pathogen disease combination for your PowerPoint or slide presentation at the end of the term. The sooner you do that, the sooner you can kind of start thinking and planning out your presentation. But you cannot just arbitrarily choose something. Those topics are pre-assigned, so message me there to get yours. I will gladly let you know, and that way, if you like to start early on things, you can get started early on your presentation. All right, that's all I have for you. Have a great rest of the week.